Not cooperative. Well, that's better. And we're still seeing these tiny eel pouts.
And all the quill worms. Haven't seen any groupings of them feeding on anything lately. Must be past lunchtime.
Okay, can come in a little bit more video. That blue.
I don't think we've had any of our ichthyologist friends and colleagues on the line today here, or in the chat room. I guess Andrea was in for a little while.
She was, yeah.
Yeah, and this is different than the one that we saw a little earlier that was also a similar shaped silvery fish.
Down about 9-0.
Copy that. Let me
that had more
get
green
a full
and blue on
specimen
it.
here video so that I can start to push out.
And Andrea, I guess, said that this is probably a silver hake, or Lucius. Thank you, Andrea, via Amanda.
When you get the opportunity, try bringing your heading to 100.
Okay. What are we testing? The 45 degrees?
That brings it in line with the ship.
Okay.
I'm just curious.
I'll try it up ahead here.
Yeah.
We're still seeing lots of quill worms.
We collected one, so we'll get up close and personal with it later today.
That move's complete. I'll wait until you guys are set up. We have plenty of time to make
I
the last
won't,
target.
but you will.
Oh, yeah?
I was
Okay,
going to say.
great.
I have a feeling you'll be staying far away.
Well, I
If
don't
we
mind
keep
looking
moving.
at them closely.
We
I just
can
don't
keep-
really want to pick them up.
If we were to stay here, we would, for the next 45 minutes, we would not make the target. That makes any sense.
Fair enough.
It's kind of two different things you're telling us there.
So we're not going to move?
No, we'll make it.
So you want us to stay here? What's that?
Squid?
What's going on over here?
Squid on the
It's
bottom.
like a squid, and that might be a different fish in the lower right.
It's funny, I was just about to say that we hadn't seen squid in a while, and I looked at camera 2, and I saw that there were lots of, what are those?
Ooh.
Are they
you
amphipods, or
tell.
what's in the water column above right now?
I
Or are those the shrimp?
really can't tell from here.
Yeah, but there's a lot more food-like organisms above us. So
Look like amphipods, maybe.
yeah, they look
Here's the
smaller
100
than
degrees.
those shrimp that we had seen. And just as I was about to say that, some squid moved into our view. They have something to eat. They did not seem to
That's
mind the
You weren't at
methane
100 though.
environments.
No.
The
You're at like
bacterial
130.
mounds, they were
I was at 100.
exploring them
Now when it
themselves.
Oh, now when it pinged? I mean, just now.
just
sort
now
of
when you were
Yeah.
burrowing
Okay.
across them. Or,
process.
Yeah, I'm not sure that that's
Yeah.
usual
It seems
behavior.
weird.
It
Right,
might
it
just
might
be,
be.
them trying to escape downward.
And you were right up, pretty much right under Sirius.
But they sure have been hitting the sedimented surface often.
No octopus today.
No.
We've seen one almost, it seems like almost every day.
Yesterday we
lately.
saw quite a few.
Oh my gosh, that made up for today, I guess.
Yeah.
And it was Octopus Friday.
Mm-hmm.
Squid Saturday.
Quick zoom here, video.
Wow.
That's different
Unusual
behavior.
posture for a shrimp.
Mm-hmm.
Hey, you guys want to move?
Yeah.
Yeah.
I'm going to do the same move.
Copy that.
220. Bridge Nav. Would like to request a ship move, please. Range 40 meters. Bearing 220. Speed decimal 2 knots. That's correct.
The crab up on the left passing.
Do you want to take a look at it?
Another one of the cancer crabs
Oh.
that we've been observing all day today, off and on. And something
Hiding
just
from me.
disappeared into that
Yes.
hole.
It's that fish from earlier.
Might be that wry mouth fish that we've been seeing, or whatever he might be. Still don't have a totally conclusive identification on him.
Don't think we're going to be able to see him. Well,
We're diving in a submarine canyon that
That
is about 30 miles
Wow.
Sure
Little
have.
landslide.
east of Pea Island on the outer banks of
Sure,
North
pilot
Carolina.
didn't take any crap from those fish.
what's that?
And this site was identified as an area with seeps, with gas hydrate or methane seeps. And so we picked this site a while ago, and through really high resolution multi-beam mapping, not only did we see the topography really well, but also looking at the water column data, we were able to see that this site had seeps, but those had been identified in the past using this vessel and then with subsequent cruises with the U.S. Geological Survey. So the area has known seeps, and particularly on the shallow areas of this submarine canyon, up in the ranges closer to 300 meters. And we're diving in the range of 500 to about 300 meters. Well, not quite that shallow today. And Cheryl, you'll be back here in a few weeks, you said, or later this year?
The end of August.
Oh, okay.
The third week in August, we will leave Woods Hole on the research vessel Atlantis and come down this way and do some
that
Alvin
shrimp
dives,
on the
submersible Alvin.
middle right is the one that we're looking for.
Okay.
Oh yeah, here comes that
Good.
shrimp.
Let's definitely take a look at this shrimp.
Let me get to the side here.
And have you been in the Alvin before?
I have had one Alvin ride way back in 2007, I think it was, in the Gulf of Mexico as
Great.
part of the Chemo 3 project. But like you, I've done most of my submersible diving in the Johnson Sea Link.
Right.
There may be another one behind it with completely buried.
I did a couple dives
Okay,
with
let's come out of this one video.
Yep.
Did you ever use that one?
No, but I've heard stories about it.
Yeah. It's the little yellow submarine.
He's only got one complete antenna.
3 of us jam-packed in there. It was a little crazy. I definitely prefer the Johnson Sea Link.
I'm gonna come forward a little bit. Get out of this.
There are great advantages to having a submersible for some purposes, and great advantages to having a tethered, remotely operated vehicle that isn't manned. And we're able to spend much more time on the seafloor
just above your laces.
with our remotely operated vehicle, and to provide instant telecommunication, telepresence, because of the nature of our tether. We have power, the ROV has power, and it can instantly relay the video feed.
That's pretty amazing behavior there.
Yeah, and it's maybe the 3rd or 4th of this species that we've seen that burrows itself. And they all have those long, long white antennae. We haven't gotten an ID on this from anyone.
Nope.
0 degrees.
Almost has eyelids. Looked like he was going to close them and bury himself.
You can come wide video.
Huh. It's amazing how
That's
long
me looking
those
right down
antennae
at you.
are.
Yeah.
A couple times, at least 2 times, maybe 3 times his body length.
Video's going to be switching out.
Okay.
Copy that. Thanks, video.
I'm gonna shake all this off.
Yep. And now I'm looking back at you, pilot. That's all the way. Had another ping on D2 just a couple seconds ago. Not sure of status.
Still see a little evidence of the bacterial mats. A lot of them, they appear to have been buried by sedimentation. So they don't appear to be very active right now. Every one where we've seen bubbles is bright white. And often some very thick matting of the bacteria.
Still looking.
And this
behind me.
darker
I saw
sediment
fire.
is
copy. I'll
underneath
push
the
up.
mats. It's a reducing environment, so no oxygen. no oxidization of the mineral matter, and so it gets very dark. Unlike the surface sediments that definitely have been exposed to oxygenated waters. These sediments have a lot of mineral matter in them, in addition to the planktonic foraminifera that settle from the surface waters. But the fine grains, some of them are being brought in probably from rivers that are off of Virginia, really. They're fine enough to be carried adrift some distances. I was actually surprised to see so much mineral matter in some of these fine grains.
Looks like you guys found some krill.
Once you get into the deeper environments of maybe 1,000 meters and well away from the continental margin, you basically see no terrigenous material. It's all planktonic foraminifera and some pteropod shells, and that's what we were seeing in most of the other environments south of here too.
Enemy on the upper right.
Yeah, we've seen a whole bunch of these guys. Thanks. If you see those shrimp, though, call those out.
Looking for shrimp?
Yeah, like ones with big white antenna.
Thank you, Bridge.
Big white what?
All right,
Antenna.
and that moves complete.
And they're on the ground. They're not like swimming.
Okay. Is that one of the lower left one? I think. With an enemy.
That crab just buried itself.
Looked like that little cancer crab just buried himself partially too.
Oh yeah, I wasn't paying attention to him. Oh, look at this one coming down the slope.
Okay. So you see this shrimp ahead
It's
of us?
amazing how fast they can move.
It's a lot
Yeah.
bigger than that.
Okay.
It's like larger than the
There's a
lasers.
There's a hag mouth at the bottom.
One of those hagfishes.
Yeah.
Didn't see it.
I'm gonna get us some room here.
Tomorrow's dive is going to be quite different. We're going to be heading north of here and then offshore to depths exceeding 1,000 meters.
Good for another move.
And it's an area where a very large submarine landslide occurred at some point in history, detaching a huge block of
Sea star, right?
rock and sediment from the steep
Bridge, are we
side
now?
of the continental margin, and that rock slid down
Maybe a
to
crab.
its current location. And Jason
much
Chater
farther
of
do
the
we have
U.S.
in this
Geologic Survey proposed this site.
Okay,
He's
can we
been
call in another
Quick zoom video.
investigating
I think it's that crab again.
submarine
40 meters,
landslides and
Bearing 220.
what their character is at
Speed
the
decimal 2 knots.
the toe of these large blocks that have detached.
That's correct. Thank you.
Submarine landslides are of great interest. If they occur with a huge volume of
A lot
material
of crabs down
that
here.
suddenly slides down a slope, it could displace water and cause some
Something there.
significant
It's a squid.
disturbance
Gray.
to the
This guy?
sea above
Gray
it,
above it.
forming a
Oh, okay. Oh,
a
new fish,
tsunami-like
maybe?
wave.
Oh, that's a mat. I think.
Matt.
But if these detached blocks just slip slowly, then that isn't as much of a hazard. And
We're also
I
looking
don't
for
actually
any
know if Jason will be
bubbles.
looking into evidence for one of those two scenarios, but it certainly is of interest to anyone living near the coast where earthquakes may cause detachment of areas of the continental margin.
Is that fish?
Another hatchet fish, I think.
Yeah, quick zoom video.
Just swam
Let's
by.
Take a look at it.
Maybe. Nope, not a hatchet fish.
We're just looking the length of the body of the barracuda.
Yeah, we were just looking right down its back, so it looked a little deeper.
All right, come on now.
There's something at the base of that anemone. I guess it's a fish.
Yeah, could we take a look at the
Looks a
base
little-
of that anemone on the left?
Absolutely.
Thank you. Wonder if this is that fish that was somewhat similar to the green eye.
Yeah. Good, he's lining up for a good view.
All right, come on in video.
Yep, that's
It
the other
is,
one.
Yep.
I wonder if this actually is a green eye.
I think it is green eye.
Yeah, you can see.
Has the right color, that's for
Yeah.
sure. Something about its head looks different. It's not as bulging eyes or squared off that I recall. But yeah, that coloration is beautiful.
It is gorgeous.
Very good.
Parasite on his tail.
Head-on shot right here.
Where have we seen them? At Lophelia Banks?
Yes.
And
at
I've
that
seen them
guy.
at the Charleston Bump, south of there off of Charleston,
I think he's watching.
associated with the Gulf Stream areas.
Wish we could see the reflection in his eyes.
Mm.
Come tilting up on him. Not sure.
Is that a parasite on it?
That's what we were checking. Not real sure.
What's that on
Yes,
its
clear.
caudal fin?
I don't know if that's a parasite.
It looks like it's something attached to him as opposed
Yeah.
to something that belongs to him. Is that a little bump on the end too?
Mm-hmm.
Another parasite maybe. Yeah, it definitely looks like a parasite or a growth.
Mm-hmm.
Wow, look at that anemone.
Do you want to see the anemone
Quite
as well?
stately.
Sure.
All right, come on in video.
Amanda suggested maybe a copepod
Let's get the
parasite.
whole
They burrow their
specimen
head
through
into
the
the
base.
fish and attach.
That's lovely.
Yeah. Thank you, Amanda. I would say a lot of parasites have horror stories.
Yeah.
Or make for good horror stories.
Let's get the tubes on the base.
All right, pilot, that's me looking down about 9-0, straight down.
Copy that. Got just a little longer here video, and then we're going to have to move.
Lots of horn tubes.
Yeah, that's quite a bumpy surface there.
Yeah, look at their little tubes. Under, you can see on the
Look on the left side of it.
upper portion, you can see that they're hollow tubes.
Oh yeah, you can see the little worm. I think it's a worm.
Yeah.
Or
All right, video, I need to move.
Substrate!
we'll take anything to get off that sediment. And Tina says she bets that these are Tennaid tubes. And Tennaids are amphipods, aren't they? Or are they isopods?
Paracarids is what.
Right.
Very lumpy
Paracarid
ahead there.
crustaceans, Amanda said. They have tiny claws.
And little brood pouches. Or a marsupium.
Thank you, Bridge.
No, I didn't realize that the giant isopod is in this group.
So that move's
Bathynomus giganticus.
Fish in a
Giganteus.
hole.
Go ahead, zoom in.
I was really hoping we were going to see
I
one of
know,
those.
everybody
Sirius
was.
Yep.
over the waypoint.
What's that? Next one, Will?
Yeah.
Is that a sponge?
I don't know. I'll get a better look. Come on out.
It's something.
There's something
that?
purple in there. I gotta sneak up on it.
Well, I'm looking forward to the possibility of rock exposure tomorrow.
Yes, I imagine so. I am too.
Yes. We are going to be climbing a very steep
Okay, come on
wall
in.
that is the edge of a block that I mentioned earlier, clearly detached from the continental slope, just east of Norfolk.
Is that like a jellyfish that has hit the deck, maybe?
It may well be, yeah. That's now that I see tentacles, yeah.
That's what it looks like.
I wonder if somebody grabbed it and pulled him into the hole. Who would do that?
You want to try a quick fog light before this dust cloud hits?
Sure. Video. You up
They
for a
may
fog
be
light?
regretting it.
Sure, we can see what it looks like.
Copy. Coming on.
No promises.
This was a
That's gotta be
Cyanea
a
jelly.
right? Yeah.
Yep, yeah.
Okay.
This is the one that we were seeing a lot of yesterday.
Video's clear.
Come on out.
The lion's mane.
Fog light.
Is that the
off.
same one?
Yeah, the lion's mane.
Where are my lasers?
Yeah, they're beautiful. We got some beautiful footage of them yesterday.
Oh, perfect.
Right
Looking
there.
up into the water column with them. Incredible colors. Black belly rosefish ducking down into one of those holes.
No interest, correct? I mean, we've seen them. Watch leads. You don't want to zoom or anything?
No, I think we've seen them.
Okay.
How about you?
Just verifying.
Thank you.
Yeah, you found some holes.
I did.
No bubbles, sorry.
No bubbles. Video. Oh, look at this guy.
And here comes a squid.
Bet he rams the ground. Oh, maybe not.
So going back to the little mud tubes that were on that Cerianthid tube, those were paracarids most likely, and in that group are the amphipods, tineids, cumacea, and isopods. And that's a group of crustaceans that are characteristic of having a brood pouch, or brood plates underneath their thorax where they can carry embryos. And some of these crustaceans even have extended parental care where they can scoop up the juveniles and put them back in the pouch later if something dangerous comes close. And mysids and lophogastrids are also paracarids.
So you're talking about the tube builders at the base of these
It's not that
tube
shrink between
anemones,
your
right?
legs, is
Yeah.
it?
The little tubes on the
On the bigger tube. Just try to clarify here.
Yeah.
Looks like a different fish coming up.
Alright, let me know when you guys are ready, and I'll put in that last
The one
move.
that's
There's
in the lasers.
There's no rush.
Yeah, maybe it's green eye.
Copy.
Not sure.
I probably at 3.5.
So is this the
He's
same
ready.
species of
Can't get
anemone
out
that
of
we
there.
saw lower down
Ready,
on the
bud?
slope, but their tubes just weren't
Yeah, go ahead.
as extended
How
as
far is the
these?
distance?
I do not know. I mean, these on the
35
anemones
meters.
we saw a little bit
35. Yeah, go for
lower.
it.
They had those brown
All right,
stripes
we'll do a
on
far
their
away zoom.
tentacles, and I
Let's try
haven't
that
seen
video.
those in the last couple that
I think
we've
we've
looked
seen
at
this
up
Okay.
here.
Yeah, definitely.
Yeah, this is the hake that we saw earlier, I think.
Copy
Silver
that.
hake.
Yep.
Full wide video.
But if we can zoom on another one of these anemones and try to look at the tube and look for the little
The one on the right.
creatures
Bridge
making
ROV nav.
the tubes on the tube,
We can absolutely do that. How about this one on the right?
looks great. Thank you.
We'll get as close to it as possible.
Okay, give me. I gotta go real
I'd
slow
like
to get to it.
request one last ship move. Range 30 meters, bearing 220, speed
Stay
decimal
full
2.
wide for me until I get up there.
That's correct.
And Tina said that she thinks that all of the tube anemones around here are going to have tenaids on their tubes.
We can turn the lasers off.
Okay, come on in video.
Thanks.
Yeah.
It's amazing
It's like
how
a,
many take advantage of this.
it's like a high rise or something.
Yeah.
All of those organisms crammed in there.
Do you want to try the fog lights here video?
Yeah, we can try it.
Yeah, those tentacles are
Alright,
different.
coming on.
Certainly from the other ones, they don't have that long, extended, thin portion.
I'm not a huge
And
fan
then
of
these
the fog lights.
tubes that they're building
I
are
like 'em.
taller
I
as
think
well.
in particular situations, they're really nice. I think in a lot of situations like this, you just sort of lose some definition.
I like it because it brings out these little worms.
It increases the
You know,
reds.
like if
Yeah.
you were to turn it off now and go back to how it was, like I think the color's better.
Yeah,
You
can we
don't get
take
as
a
much
peek in
blurriness
the
'cause you don't
tentacle
have all the
area again
backscatter.
at the mouth?
Absolutely. Oh, he just caught something.
They do have stripes on them, though.
Oh yeah, they do.
Oh, look, he's capturing something right now. 2 amphipods.
Looks like it.
Here comes another to the rescue. Nope.
That was a bold
Decided it
move.
was better left alone.
Video, let us know if you'd like to try without the fog lights as well.
I think we're clear on this.
Okay.
Fog lights are coming off.
Tentacles are beautiful.
Are you happy with that one, watch leads? Should we move on from?
That was great. Thank you.
Okay, heading up.
I guess we have a
So
little more
you're
bacterial
almost
mat
on
maybe on the right side.
3.6.
just down
Once
from the
you're on
anemone.
waypoint
Yep.
6, let's
Just a little bit,
this
little
guy.
hint of it.
You want to do it? It's going to waypoint 6. Go ahead, watch lead. No, we have not this shift. Great. Okay, EX, correct?
We're
We'd like our front row to introduce themselves, please.
Yes, currently sitting pilot is Andy Lister, and over to my left in navigator
Is Josh Carlson.
Sitting as co-pilot is Sean Kennison.
And over on the far right is Roland Bryan doing video.
And in the back row, Bob Knott clipping.
and we are all with the Global Foundation of Ocean Exploration.
And what a fantastic team to work with. We've really enjoyed working with you for the past 3 weeks or so on this expedition.
Hydraulics coming up.
And all of your hard work really pays off for us and all the other scientists who are using this information and the public, so that we can bring these beautiful organisms to
zoom
their
in video.
computers or their
We're going to lose it in a
TVs,
second.
Resetting the
smartphones.
skin and firing.
Stream 3, I've got the Niskin bottles so you can see them close.
Watch lead, this is Nav. We just fired.
Yeah, we're
the
about
last
to close some
Niskin
Niskin
bottle.
bottles.
Great,
If you
thanks
want to look
very
at
much.
Stream
Do you have
3,
the spec number for
you'll
that?
see it in action.
Spec 7.
Spec 7.
So these are
Copy.
water sample collection
Thanks, video.
devices, and we're going to send a signal down, and they will close at each
All right,
end
we got to
to
find one of these
capture
shrimps.
about a 2-liter
1.7
quantity of
liters.
water.
Yep.
And we've been collecting them at several depths on today's dive. And that water will be used for chemical analyses and also environmental DNA analyses.
Fun fact, those were designed by our navigator,
Mr. Niskin.
Mr. Niskin himself.
Thank you, Bridge.
Sitting in nav, designed those watch leads.
Designed them on so that they would release on the ROV?
Yes.
Oh,
on
okay.
these, yes, our
Okay.
particular.
Long-legged
I didn't think his
ship,
last name was
black
Niskin.
laser.
No.
And he's awfully young.
I did not design the bottles. I designed
Our
the
particular
release mechanism.
Release.
Well, that's really great. Thank you. We appreciate having them available
clear.
to us.
Coming off.
Come on in video. It's going to be a little dust storm here, unfortunately. Came at him at the wrong angle.
So watch
down.
lead,
This is the
Nav.
other type of shrimp we've seen without those long white antennae. Very delicate legs. So far we have not seen this one bury itself like the other species,
Watch lead, this is Nav.
who has yet to be identified.
So we're
It's a
at
dramatic
waypoint
face shot
6,
there.
and we have about 12 minutes left in the dive.
Come on now, video.
Would you like
We
us
got
to
this one
continue
earlier. We're looking
going,
for that one with the
uphill?
really long white antenna.
Yeah, good copy.
Wait, what is that? No, nothing. Never mind.
And so we've completed our dive track.
Coming back around.
Now we have a
co-pilot.
few minutes left, and so we'll just continue up
Copy.
slope here and
Well, that
see how
timing
far we
was
get.
pretty good.
Yeah.
Good job, Derek, our mapping lead. Plotted this out. He knew exactly how far we could get in the course of this dive. And speaking of Derek, he's not on the microphone, but Derek Sowers is to our left. He's lead of the mapping team. We have a great group along with Kevin Jerem, who does the night watch.
Watches, do you want to look at this one as well, just to see, or are we happy with these anemone?
Oh, we could take a look. Might as well. We have time.
We do. I will land.
And
Shifting.
Tina pointed out that
Laser's clear.
Pachycerianthus multiplicatus from Ireland has more than 40 species of associated fauna, including crustaceans, polychaetes, and sipunculids.
Go ahead, video.
Tilt down.
And there's a species of Pheronus that lives exclusively
Look at
on
that guy.
cerianthid tubes. It's something that I never even appreciated, that how much habitat
little-
these guys create.
A brachiopod,
Oh, it does look like a brachiopod.
the little shell-like thing on the bottom. It's hard to tell if that's 2 valves or if it's an open cup. We have the tube of one of the, the quill worms standing up next to the base. I wonder if anybody's home or if
Yeah.
how it got oriented in that
Tilt
direction.
up.
And it looks like this Cereanthid has eaten another salp. Is eating it.
Is
We're
eating it.
watching it
Yeah.
happen live.
It looks like eyes. They're looking at us.
It's a little face there.
Yeah.
Video's clear.
Okay. Come on out. Come on.
And then now we could probably get another move in.
We're
But,
seeing a
sitting
lot of
pretty
those
at
little
45
pinhole
degrees in my camera.
burrows.
Mm-hmm.
Good copy.
Thanks, Amanda, for noting that, because we really haven't seen those until now. A couple days ago, we took a scoop sample of
before is on
sediment
the
that
right.
had these, and we came up with at least
Bridge
10
area
acorn worms in the sample.
Go ahead.
And they burrow down into the sediment and make those little
We
holes.
would like to request a ship move.
Oh, get
Range
sediment tea.
And I would
40
just as
meters,
soon not make
bearing
another collection of them.
220. Nuts. That's correct.
And Tina points out that there's another species of
Another squid back there.
bivalve that's an obligate
Really
symbiont
large
on
Yeah.
Cereanthid tubes.
Go with lasers back on.
Yeah.
Sure. Go for it.
And that's a tropical species.
Oh, he does not like lasers. He's getting
There's
ready.
the-
This is probably one of the
You have a
elex
quick zoom here video if
squids.
you'd like.
yeah, leaning on his elbows.
Lasers are clear. Oh, there we go. Oh, I'm not in a great position to stop.
Our colleague Stephanie Bush narrated a great short video that is now on the website
Okay.
for this expedition, discussing the chromatophores of these squid. So it's a great little educational and interesting piece if you're interested in that.
This guy. He's always on a rampage. Is this a buried tube anemone, maybe?
Yeah, it looks like it.
Quick zoom video.
Maybe that's has the stripes like the.
Well, the inner tentacles of the tall ones had stripes also. Yeah, we'll have to compare the screenshots
Dust is
later.
coming
Mm-hmm. And that one looked like a very short tube compared to the others that we've
Yeah.
seen. If it had a tube at all.
And while we are nearing the end of this dive, we want to say
I'm
thank
gonna
you
bury
so
it
much
with
to
that
those of
move
you who
back.
have given us a portion of your Saturday on the chat room
Quick
to
zoom
help us
here
out
video.
with identifications and discussions and interesting stories, as always.
Actually, it's
There's another flatfish at the base of our view.
I'm going to bury him, I already know.
We've seen a few of these today.
Okay, great.
Different orientation, so we might get a different lighting on it to help it in ID.
Go ahead, video, if you want.
But thanks to Amanda Demopoulos from the USGS, Andrea Quattrini from Harvey Mudd College, Asako Matsumoto from the Planetary Exploration Research Center in Japan, Bernard Ball from Duke University Marine Lab at University College Dublin.
Both. Yeah.
Oh,
he
I
has
see.
two affiliations, I guess.
I didn't know that Duke had a marine lab in Dublin. That's why I was surprised. Well, aren't you lucky to have both of those affiliations?
Yeah.
And of course, Carolyn Ruppel, who helped us a lot today with her calling in and giving us information about the seeps, and she's at the U.S. Geological Survey in Woods Hole.
There you go.
Daniel Wagner from NOAA OER was hosting a group at the South Carolina Aquarium and wrote in for some of the questions from their audience. Eric Cordes from Temple University. James Murphy of NOAA OER, he's the Hawaii Sea Grant Knaus Fellow. Jason Chater from USGS in Woods Hole, and Jenna Hill, also from U.S. Geological Survey. Kate Rose from NOAA NCEI. Nolan Barrett, Natural Products Discovery Lab at Medical University of South Carolina. Bob Carney from Louisiana State University. Scott France, University of Louisiana at Lafayette. Scott, this is one of the first days he hasn't actually called in. We've missed your chat, but
yeah, really,
or your voice.
There's still a few minutes left, Scott.
We certainly had a lot of chat from you, typed instead of verbal. But thanks for helping out. And Scott is also the science advisor for all of these Atlantic expeditions under the heading of Aspire. Scott Harris from the College of Charleston. And Tina Molodtsova from the Shirshov Institute of Oceanology in Moscow. And Zach Prue, who is from College of Charleston. He's my graduate student. And he today was at the visualizations or the Viz Lab at the college and hosted several visitors.
That striped fish looks like a green eye.
So I hope I didn't leave anyone out. Did you see anyone else who I missed?
Did you get Bob Carney?
I did.
Okay.
I did.
Jenna Hill?
Quick
yes.
zoom for a second.
And
oh, yep. Good eyes.
I don't have, my whole list hasn't been showing for some reason, so.
Oh, yeah, we have that problem with our chat room feed sometimes.
Fish in the midwater?
Yeah, let's go for it.
And we were hoping to see a little more seepage.
Oh, no, he's laying on the ground.
Yes.
As we moved
We've
higher
seen
up
this guy.
onto this
Quite
slope.
crazy.
But we certainly saw some good examples farther down.
Yeah, very extensive bacterial mats.
Yeah, somewhere in the range of 400 meters plus or minus. We didn't write it down as we should have, but certainly it's on the video.
Mm-hmm.
Saw some very extensive bacterial mats and areas of bubbles. So some good validation of the remotely sensed data. But unfortunately, no chemosynthetic macrofauna.
Yeah.
There certainly was the microfauna, the bacterial mats, and probably microbes living within those. We saw some tubes that might have been produced by the microbes.
About a minute and a half.
And as Amanda Demopoulos' research has shown, there's probably a high diversity of infauna or animals that live within the sediments underneath those bacterial mats.
Let's find something great.
It's your last chance, Andy.
But we didn't have a good
I'm gonna find a whale.
good way to sample those on this vehicle.
Dream big.
I can zoom you out into the distance.
Yep, I see one way out there. Just need to get enough time to get there.
I think this is the clearest it's been out there.
It definitely is.
I can actually make out. Yeah, just like we practiced.
Okay. On the talent show.
We've seen mud stars, and that's about the only echinoderm today. See one in the distance there.
Okay, second one up there.
Just a few crab species dominated by cancer.
Is that a ridge?
The genus cancer.
The genus cancer, yes. Sorry.
The crabs don't have cancer.
No.
All right, Andy.
Another black belly
That's about
rosefish.
it.
We've
It's right
seen
behind
some
that dust
great
cloud.
feeding behavior today.
All right.
That's for sure. And I'm not
Alright,
sure that
we'll
I
start setting
quoted
up,
Amanda's research correctly. It's a high abundance of infauna
All your
in the
lights
mats.
flash, okay with that?
Lots
Yep,
of
go
numbers,
for
but
it.
only a few species. So the diversity is low, but the abundance is high.
Ooh.
Yeah, that would make sense, wouldn't it, in
That's
sort
what
of
it
an
looked like
extreme
earlier.
environment.
So
Yeah,
we'll
you
back
wouldn't
from
have a lot of
yesterday.
species that could tolerate that.
Right. We see that hydrothermal vents as well.
I'm gonna head back towards you.
No, that
Shoulder.
thing's kind of new. What is that?
Salps.
Oh,
Yep, some salps in the water column, and if it's anything like yesterday, there's probably going to be a lot of organisms in the water column as we ascend at the end of this dive. We're getting ready for that now.
Kind of off screen now.
Yep. I'll come back. I was gonna turn and look at you to set up.
Copy.
Yeah, kind of toxic environments, high
can
in
turn.
sulfide and
Return. I didn't get your
inhospitable
uppers yet.
mud.
Oh, okay. No, I
Glad
mean,
we
you
don't
can
have to
turn
live there.
face
Oh,
me.
turn
Yep.
to face
Yeah,
me.
yeah.
Copy.
It's gonna be that same direction.
All right, great. We got visual. Start with your uppers.
so on recovery, we're just going to have the ship come ahead.
Perfect.
So, and it goes into deeper water.
How much vertical and horizontal distance have we covered today?
I'm not sure, Roland.
Check
All of it.
that a little bit later after we're not in recovery mode. What's that? Go up.
Amanda concluded with, it's probably smelly because of all those sulfides, but I doubt the worms care.
Shilling looks good to me. I'd say,
How's craft look? Looks okay.
yeah.
Have you reset the Niskin pin?
We get one more
This
view
one
of our
is
last
Reset.
hagfish. I think that's a hagfish.
We can do a zoom while we
Distance.
set up here video.
I
Yeah.
didn't quite
Suggest you
see
turn
it.
to
Right
your
there.
port and tilt all the way up and zoom in. You'll find the well.
Oh, yeah.
I already saw that one. So
It has a cable attached to it.
Leashed.
Oh, yeah, there's a,
ready?
is that a
And
bigger
lights
eel
just
pout
up
on the
out.
right?
Okay, if you want to turn,
No, I don't think
Or
that's
could,
an
could
eel
be that,
pout.
that
Yeah,
hake,
the
the
hake.
southern
and you
hake.
can come up a
Mhm.
little bit.
I
I'm going
think
to go
so.
underneath you.
Copy. I'll just center you then. Is that all right?
That's perfect.
Great.
And just give me just a little. Yep.
Coming up.
I'm going to come underneath you here.
Yep. Delta about 16.
That's great for me to come underneath you. You want to continue turning? So you're-
Yeah, I'll line up with ship.
Yep, and I'll.
It's a beautiful ballet you guys are doing right now.
Thank you. We practice this.
Very nicely done.
Trying to get less than 7 turns.
That's the goal.
Okay, I'm pushing out. I still got visual on the ground.
Copy. I'm
The
lined up
pilot's
with
doing the
ship.
last maneuvers before
Good delta.
lifting off the seabed.
Okay, going under you.
Copy
With
that.
one parting shot
That's
of a
me
crab.
looking
Seems
all
like
the way
almost
back.
all of our dives end with a shot of a crab. The spider crab this time. Oh, and there's cancer crabs also making their last show. Wow.
Got you in tail
And we're
cam.
going up through a dense group of organisms that you can see on camera 2 right now. But we'll be lifting up through it shortly. And we think these are